We define epistasis as the expression of alleles at one locus that masks or modifies the expression of other alleles located on a different locus. Pictured below in the graph are the different kinds of epistasis, with its designated ratio, genotype orientation, and type of interaction associated with the type of epistasis. Now that we know what an epistasis is, we also need to know what a locus is. We define a locus as being the physical position on a chromosome where the gene is located. In this video, I'll be discussing how the black coat gene and the chocolate coat gene interact to determine the phenotypic trait of a cinnamon coat color, which will be looked at in a typical household cat. There are 6 different types of epistasis, but for the purpose of this video, I will only be discussing single recessive epistasis in a household cat using a pigmentation pathway for my example. In the following pathway, the big letter A represents the black coat gene, and the big letter B represents the chocolate coat gene. Though capitalized A and B represent the dominant alleles, and lowercase a and b represent the recessive alleles in this pathway. In the drawing, if any form of a dominant A allele or a dominant B allele is expressed, no matter if it represents a homozygous or heterozygous genotype, it will allow for both enzyme A and B to be produced and thus allow for continuation down the pathway, resulting in a cinnamon coat color phenotype. If we have a household cat who shows any dominant A allele in its genotype, but is homozygous recessive for the B gene, this will lead to the production of enzyme A but inhibit production of enzyme B due to recessive B alleles. This pathway results in a household cat expression in chocolate coat color phenotype. Additionally, if we have a household cat who expresses homozygous recessive A genotype, but 2 has a dominant B allele in this genotype, enzyme A will be masked, which halts continuation down the pathway at the first locus. This results in the expression of the black coat color phenotype. The same information applies if a household cat represents both homozygous recessive A and B alleles in its genotype, where again enzyme A is masked, resulting in expression of the black coat color phenotype. With our understanding of this pathway, we can start to discuss the element of a dihybrid cross, where 2 heterozygous parental parents will create different phenotypic offspring. This cross will help determine both the genotypes and phenotypes of offspring produced. Using the table provided, we can see that 9 offspring who are dominant in both the A and B alleles produce both enzymes that results in a cinnamon coat color phenotype. 3 offspring represent a dominant genotype for the A gene, but homozygous recessive genotype for the B gene, resulting in enzyme B not being produced and a chocolate coat color being expressed. Lastly, 4 offspring represent a black coat color phenotype due to these individuals expressing the homozygous recessive genotype of the A gene, resulting in enzyme A not being produced and inability to continue down the pathway. The result of this phenotypic ratio is 9 to 3 to 4, which is expected from the dihybrid cross and correlates to single recessive epistasis. The last cross we will be discussing is between one parent who has a phenotype of chocolate coat color and one parent who has a phenotype of black coat color. Considering that this cross is not a dihybrid cross, we should not expect this example to be associated with single recessive epistasis. In the table, you can see that 3 coat color phenotypes arise from this cross, which are black, chocolate, and cinnamon. 4 cinnamon coat color cats arise from the dominant A and B alleles being expressed, allowing for both enzyme A and B to be produced, resulting in this phenotype. Additionally, 4 chocolate coat color offspring arise because of their dominant A alleles at the first locus, but 2 expressing a homozygous recessive genotype in the second locus, inhibiting enzyme B and resulting in this phenotype. Lastly, the 8 black coat color offspring who express the homozygous recessive genotype at the A gene cause inhibition of enzyme A, resulting in this phenotype. This cross leaves us with a 1 to 1 to 2 ratio. Thank you for watching this video, and I hope this has helped with your understanding of epistasis.